My name is Ola Hammarsten. I'm a professor and chief physician at Clinical Chemistry Sahlgrenska University Hospital in Sweden. In this video, I will propose that release of troponin from unstressed cardiomyocytes is a more or less normal phenomenon and not restricted to dying cardiomyocytes. I will also propose that most released troponin is taken up locally by cardiomyocytes before it reaches circulation, a system I call cleaning your own backyard. If this local clearance of troponin is impeded, this will result in stable troponin elevations. The local troponin clearance is likely due to scavenger receptor-mediated uptake, a system called extracellular proteostasis. Impaired proteostasis is tightly linked to aging. Stable troponin elevations might be a sign of impaired removal of sticky unfolded proteins between cardiomyocytes. In this light, stable troponin elevations might be viewed as a heart-specific age marker and thus explaining why stable troponin levels are so strongly linked to heart disease and death. This project started when we found surprisingly high levels of troponin in the culture medium growing different types of cardiomyocytes. Initially, we thought that this was due to dying cells, but we find no evidence for that explanation. Macromolecular exchange via holes in the plasma membrane without cardiomyocyte death is possible in part because the cytoplasm is a gel with restricted diffusion. However, the most important protection against cell death when the plasma membrane is injured is a calcium-dependent repair system called cell wound repair, discovered over 90 years ago. The result of cell wound repair is likely formation of a membrane patch within a few seconds by vesicles fusing with the plasma membrane in response to massive calcium influx. Based on the rate of troponin release from cardiomyocyte monolayers, we made a constant intravenous infusion of the amount of troponin that would be released per minute from a normal 1 gram rat heart. The constant infusion of cardiac extract resulted in several hundredfold higher troponin levels than we observe in normal rats. Then why do rats have only low levels of troponin in their blood? One possibility is that troponin is cleared by surrounding cells, cleaning your own backyard. According to this hypothesis, only a small fraction of released troponin ever reaches the circulation because of a local uptake by cells in the surrounding cardiac tissue. Using fluorescently labeled troponin, we observe robust endocytosis of troponin by primary rat cardiomyocytes that is blocked if cells are incubated at +4 degrees, a classical control in endocytosis studies. Uptake of troponin is likely mediated by scavenger receptors known to be expressed in cardiac tissue. After intramuscular injection in rats, very little of the injected troponin reached the circulation, consistent with the local uptake of troponin by muscle cells, known to have a very active endocytosis system. Hence, a troponin molecule in between quadriceps muscle cells is unlikely to reach the circulation, at least in the rat. Based on our preliminary data, we proposed that proteins that leak from cells in the 3D context of a tissue, for the most part, are cleared locally by the cells themselves. The hypothesis dictates that local clearance will not be very efficient if cells are grown in 2D monolayers, but much more efficient if cells are crammed close together in the context of a 3D tissue as shown here. In a separate movie, I will speculate on how local clearance and cell wounds might explain several features of both transient and stable troponin elevations.